Good evening, everyone. It's a pleasure to have you all at the second session of our ongoing webinar series called Precision Health 101. And before I introduce you to our speaker today, Ms. Elika Garg, just keeping things, I have muted you all. So if you have any questions, please type in the chat box, or you can send the question directly to me and Elika, and we can answer them at the end of the session. So without further ado, I'll welcome Elika Garg, who is the founding member of wellOwise and chief bioinformatician at wellOwise. She is a gold medalist from NIT Wallonger and has a master's in human genetics from McGill, Canada. Prior to joining wellOwise, she was part of the group at the Ludmer Institute of Neuroinformatics in Canada, where she and her group developed the first PRS risk assessment model and software. She has published in BMC Bioinformatics, PNS, and Nature Communications. Her work at wellOwise has been instrumental, and she has been using her knowledge and skills in genomics to develop the precision health products at wellOwise. So over to Elika Garg.
Hi, everybody. Okay, let's start my presentation now. Okay, welcome. I want to show you how we use genetic information at wellOwise to assess disease risk and provide a plan to improve your health status. Since I'm in charge of the bioinformatics analysis, I will go into a little bit of detail about how we extract useful information from over 500,000 mutations that our data provides. Our health is shaped by multiple factors, but at the core of it is our genetic architecture that we inherit from our parents. As I wrote in the blog, you can imagine this being the scaffolding of your house, the steelwork and frames, the beams and ceilings, which is more or less a rigid structure. However, a much more flexible element shaping our health is our environment, which can be anything from your exercise regime to your smoking habit, your dietary preferences to your resting state. This is the paint and furniture in your house that you can move around to achieve a better outlook. In tandem, they define your health, genes and environment together. For this talk, my focus will be on the genetics. So let me first show you the kind of genetic data we analyze using the Illumina's global screening array microarray to get genotyping data of over 500,000 SNPs. That's 6.5 locations in your DNA spread over 22 chromosomes, the X and Y chromosome, as well as mitochondrial DNA. 6.5 lakh is still a fraction of the whole set. But since these locations are selected because of their importance, they can point us to problem areas. Now, some diseases following Mendelian patterns are heavily influenced by a single gene or even a single variant. Any disruption in that one single gene or SNP, and you may get the disease, for example, sickle cell anemia. But other diseases are more complex. They follow a polygenic model. Cumulative disruptions in multiple genes across chromosomes can influence them. For example, diabetes or asthma. They are more prevalent in the population. They operate on a continuous spectrum and can have chronic disease status. But they are also more susceptible to environmental effects. You may be pre-diabetic because of your genes, but if you follow specific nutritional plans to correct it, you may be able to prevent full-blown diabetes. How can we measure this risk of polygenic diseases? We can use a polygenic risk score, which is a summarized number adding the effects of all SNPs. If a SNP is more important for a disease, it will have more weight in the calculation. When we plot these scores for enough people, we get a normal distribution, as you see in the population plot. Controls are people who don't have the disease, and their distribution is aligned to the population. Cases are people who have the disease, and their distribution will be shifted to the right. That is, they will have higher scores than controls. We can use this knowledge to stratify risk. Blue is baseline risk. Such people have average risk, average amount of risk. Next, yellow is minor risk, people with elevated risk profiles. And red is for major risk, people who are at the extreme end of the highest scores, most likely to develop the disease. For this PRS calculation, I mentioned SNP weights are used. But how do we get them? How do we know SNP A is 2 times more important than SNP B for a disease? We gather this information from GWAS's, genome-wide association studies. However, these studies are limited. Because the SNP weights will differ for every disease, and not all diseases are covered by GWAS's. They are expensive. Secondly, most of these the studies are based on Caucasian populations, which means SNP weights can be very different for non-Caucasians, for example, people of Indian ethnicities. Similarly, both genders are usually considered together, but we know that there are gender differences in disease manifestations, and these are not always captured. So at wellOwise, we have devised a GWAS-free approach to increase the number of diseases we can offer, and we also use Indian ethnicity population controls, along with gender-based comparisons. For our standard consumer panel, we use over 1 lakh SNPs spread across 4,000 genes covering more than 150 traits in 4 categories of health, wellness, fitness, and nutrition. Okay, so for this part, let me show you our internal dashboard for one person. Okay, I have preloaded this dashboard for one person, and we'll look at it live, how what kind of information we have access to. This is the genetic analysis dashboard. This is our consumer panel summary. You can see that for this person, they have 8 major risks, 23 minor risks, and the other are baseline risks. Okay, we're gonna view it. All right, so the 4 categories that I mentioned: fitness, health, nutrition, and wellness. And you can see how their major minor risks are distributed among them. Can you see my mouse pointer? There?
Yes, we can see.
Okay. Okay, so let's go into fitness first. Just wanted to show you the subcategories. So in fitness, we have these 3 subcategories. Okay. Wellness. Wellness, we have 1, 2, 3, 4, 5, 6 subcategories.
And Elika, it's worth mentioning here that within each of these categories, you can zoom in further. So within each categories, for example, allergic diseases, you can further in the category below, if you click on the left-hand side below.
Let me show that for nutrition, Saher.
Okay.
Yeah, I'll just quickly show the health subcategories as well first. So the health is our biggest category. We have bone, cardiovascular, dental, ear, eye, you name it, we have it. And then nutrition, where I will go into more details. So here, Okay, let me pick food allergy because this person has 2 minor risks for it. Okay, in food allergy, fructose intolerance and shrimp allergy are the minor risk for this person.
Elika, you can even show that how we can go within fructose intolerance and dissect which SNPs are actually responsible. So here on the top, you can show the SNPs. So if you click on fructose intolerance, highlight that, then you can even look at which SNPs are responsible for fructose intolerance. What based on what PRS scores as compared to the population this person has been picked for this particular trait.
Well, it might take time.
Yeah, okay, yeah. Let's go ahead, yes.
Yeah. Okay, I just want to quickly show the minerals and vitamins too, because they're easier to see what actionable insights you can gather from them.
Sure.
Hello. Okay, here we go. Okay, so for minerals, let's see calcium, iron, magnesium, potassium. And for this person, if you see iron level, we have 4, and all 4 have some amount of risk. Like here. So for this person, this seems important in the minerals category. And vitamins, we have all the different vitamin levels. And you can see that vitamin B12 and vitamin B6 deficiencies are risky for this person. So this person might need additional supplements for these 2 vitamins.
Also, I mean, it's worth mentioning here that all these parameters are only the sort of the genetic scores, and we combine this with the actual blood data, the lifestyle data. So
The
we would know
environmental.
from their symptoms as well. We would know from their blood report as well what their B12 levels is. And this is very important so that we can relate the findings that we find here. We can relate and correlate them with the lifestyle and the blood marker data. So it gives us more authenticity to validate our findings from the genomic PRS models.
Also, maybe they're already taking a lot of food, different food that are rich in vitamin B12, so they might not need so much additional supplements after that.
Exactly. But predominantly, a lot of vegetarian Indians are prone to these allergies, and we have seen it across our consumers that the vitamin B12 in particular has been deficient in a lot of Indians who are predominantly vegetarians. So it's quite an interesting finding in itself, I suppose.
Okay. I wanted to show you the gene network that we construct for a person. We find all the different genes covered in this panel. So I mentioned more than 4,000 genes. So all those 4,000 genes. And we find the most important ones for this person. And we use StringDB to construct these networks that show us how the different genes are connected and which are the central nodes. Like, for example, here, this one is pretty central. And some other, like for 2 here, only has 2 connections.
So, I mean, in gene network as well, Elika, as you pointed out, FRUCT2 and IL2, where FRUCT2 is an allergic, response to fructose, and that has come up, and the person has shown to be fructose intolerant. So it is quite an important gene. Again, HLA, DRA is a gene implicated in immune responses. And again, this person is allergic, and we are picking those up. LARC2 is implicated in Parkinson's, and we picked up the risk. So it's safe to say that, the gene network very clearly gives an essence or a flavor, overall flavor of what is going on, what is at a genomic level, at a gene network level is going wrong in this person. And then we can dissect each gene and go and look into that.
Yeah, it tells us which pathways are implicated through these genes.
Exactly.
Yeah, StringDB also gives us this nice functional enrichment table, which has KEGG and gene ontology parameters, keywords. We can also go into details with that. But let's first see what kind of details we have for a particular gene. So here, IL-10, which was picked up in all of these categories. Let's look at this. IL-10 is interleukin 10, and we have some details for it. We get data from DB NSFP, we get function, disease, we get interest summary. We also embed some data from GeneCard. And this is entries is fresh pull, so it's so you can see May 2020. This is like pretty recent annotation for IL-10. Apart from that, we also get data from CPIC, Clinical Pharmacogenetics Implementation Consortium, as well as DGI, Drug Gene Interaction Database. CPIC has fewer genes, so this one is not available in CPIC, but DGI does have IL-10, so let's see IL-10. Okay, so first of all, we get these categories that IL-10 falls under. Now it's clinically actionable, the growth factor, and then we can see all the different drugs associated with IL-10 in DGI, and it also gives the source for that particular drug gene interaction.
And also an interaction type, so whether it's an inhibitor, whether it's a,
Yes,
what sort of a role it's doing as well, which is quite
if
helpful for
that's
the
available,
clinicians.
right?
Yeah. I mean, for this IL-10, the CPEC data is not available, but through CPEC data, we can also see how well a person is metabolizing particular. Hello, I think somebody.
Right. Okay. And yeah, this one is a nice feature. This is just to get the whole list in one place, the 8 major risks and the other minor risks. We can look at the entire list at one go. And maybe let's say if I wanted to just see deficiency. Okay, so all the different deficiencies. And also, right, if a person has family members in our database, then we can also look at the comparison of the different risks between their family members. And these are de-identified, so that's not a problem. You can see how the different traits go in. So fatty liver disease, this person, our primary patient here has minor risk, but 2 of their family members also have major and minor risk. And if we look at all, see that how many different diseases this family altogether has risks for. 90 different traits. And apart from that, if we want to look at a particular SNP or disease, we can also do that. We can just go into details for that. And finally, we save all of this information and go back to our platform where we combine that with the environmental data before we generate the reports. And we use nutritional databases to help us formulate diet plans for our consumers. Okay. Let's go back to the presentation. Right. If you're interested in our program, you can sign up on the platform. You can give us your health details, and the environmental factors that we keep mentioning. We'll collect your blood sample and run it through the platform that I just showed you, the genomic analysis pipelines, combine it with the environmental data, the layers of it, and give you personalized recommendations and diet plans. You can also track your health parameters and get free follow-ups. That's wellOwise. Before I wrap up, just wanted to mention that our app is available both on Android and iPhone. And you don't need to purchase a plan right away. We offer a free assessment based just on the environmental data. So do check it out. And thank you for listening.
Great. Thank you, Elika. Thanks a lot for the presentation. I welcome any questions from all of you. And we already have one question for you, Elika, here. So I think Shikha is asking, how are you assessing risk for an individual? Do you do whole genome sequencing for each person?
Genotyping, whole genome is more expensive, so people usually don't prefer that. We do genotyping, and like I mentioned, it covers more than 6.5 lakh locations in your DNA. And we then combine that information. We use the polygenic risk scoring. And for our consumer panel, we have a pre-selected list of more than 150 traits. And this is, we can change this up. We can customize this list for certain consumers or other businesses to change that according to their preference, which one, if they want to expand a certain category, if they want to expand nutrition, let's say, want more focus on that, we can do that.
Yeah, also, I mean, it's worth mentioning here that for the consumer testing, GSA, Illumina GSA is quite a good tool. But for future work, we are also thinking of personalizing it, because a lot of pharmacogenetics SNPs are not currently available on GSA, and a lot of disease-associated SNPs that are important for Indian ethnicities are not currently present on GSA. So for most of the consumer work, we focus on microarrays, but for our clinical work, we do both exome and whole genome sequencing. I have another question for you, Elika. How do you shortlist the traits? How do you find relevant genes for Indian population? Are they different from Caucasian population? And this question is from Dr. Pramod Singh.
Okay, let me first answer the gene question. They are not Indian-specific genes, but the variants. So we assign weights to the variants based on how important they are in the genetic. And because we have this information from our ethnicity controls, we kind of know that, okay, if a particular SNP is in all the Indians and it's labeled as pathogenic, let's say, for Caucasian populations, then we can kind of reduce its weight, because if it's in everybody and nobody has that disease, then it's not that important for Indian ethnicity. So the genes are not ethnicity specific in our analysis. The SNPs are. What was the first question again?
How do you shortlist the traits?
Okay, the traits were, well, we picked these traits, these 150, 160 traits based on what we thought. First of all, they had to be, because our focus was on chronic diseases. So we were looking for those kind of traits, chronic diseases, polygenic traits, and the ones that are prevalent in Indian population that we can also provide actionable insights for. So those were some of the criteria that went into the pre-selection of these traits. But like I said, this list is flexible. We can change it. We can increase the focus on a certain area.
I mean, I would say for the consumer panel, the shortlist of the 200 or 180 traits that you see has been with the particular focus on what chronic diseases are highly prevalent in India, what nutritional deficiencies are highly prevalent in India. Those key parameters were there. We also focused on looking at the traits that were truly polygenic. So we kind of did not included a lot of allergies or things that were controlled by a single gene, or much information was not available. So we did not included those traits. So those were the two major criteria of shortlisting the traits.
And also from the bioinformatics perspective, you remember I showed you guys this and here the bell curve you see. So we made sure that the diseases we picked did in fact have population distribution, a normal distribution for populations.
Yeah. So for I think initially we had like list of 500 traits, but for the ones that were sure normally distributed,
we whittled down.
Came down to 200 traits. Okay. The next question is again from Shikha, and she's asking, for free assessment, what kind of environmental factors do you consider?
I think it's like the ones that I mentioned here. Like, what's your BMI? Do you smoke? Do you drink? What are your dietary habits? And these kinds of environmental data. Also, your family history. That's very important for our phenotypic data. Claire can add in.
Yeah, I can add to this. So, the free assessment or the lifestyle assessment that we ask you on our app or the dashboard if you join free is based on a very detailed assessment that combines readily available. We combine almost 20 health assessments that are published in literature for chronic diseases. We combine all those, and it's asks questions basically about your habits, about your diet, your symptoms, any current symptoms you have, and family history. So we combine all this and make this into a 20 very easy to answer interactive questions that you can take on the app or the dashboard. So there is another question for you, Elika, from Aditi, and how it is different from traditional risk analysis platform first. And the second part of the question is, are the lifetime risk for a fixed time frame, 5 or 6 years? I think she's trying to ask how it's different from the traditional risk assessment platform, and are you able to assign, are these risks for lifetime or these change over time? That's what she's trying to ask.
Okay.
Am I right, Aditi? Okay, so you can ask a follow-up question if I've not asked your question correctly.
Okay, so let me just the first question, how it's different. So because as I showed you here, we focus on a polygenic approach, so it's not just
Elika, can you put it on the full screen? Yeah, it will be better.
Right. Okay, yeah, so we do polygenic approach. We don't just focus on single genes or a few variants or a few genes, like 10 genes or so. We go wide. We look at multiple different genes to see how together they add up your risk for a certain disease. So that's different from the usual offerings in India. And also the ethnic controls as well as the gender analysis, that also plays a major role in our risk assessment, which may not always be available in the reports of other companies. The second question was?
The second question was, are these risks a lifetime risk, or is it for a fixed time frame?
Well, they are lifetime. The genetic risk is lifetime. You can get this test done for infants, and their DNA will not change so much that well, there would always be somatic mutations, but most of the risk, especially because this is PRS-based risk, so they will remain more or less constant. So if you remember, I mentioned that your genetics is like the architecture, is the scaffolding of your house, which is more or less rigid. But the environment that I mentioned, that you can modify. So your lifetime risk will also be dependent on the environment. So if you keep on eating sugary foods for like 10 years of your life, your environmental risk for diabetes would increase. But then if you cut it out and go back on a healthy diet, it'll go back to being better, and your genetics won't really play a big role there because it's all about your environment in that particular scenario.
Yeah. So I mean, it's very important. Sorry, it's very important to point out here that all these traits that we are talking about. One thing we have to understand that you're talking about complex traits. So there is almost like a 50-50, if not a 30-70 contribution from lifestyle as well. So the scores that we see here, they could be a minor risk, and the minor and major risk are very fluid, so to say, depending upon how heritable that particular trait is.
Yeah.
That we don't talk about. For example, for diabetes, the heritability can be something between 30 to 50%, whereas for a neuropsychiatric disorder like depression or bipolarness, it can be much higher to 70 to 90%. And that is very much kind of the scores would not change, but your environment has the power to change your predisposition or change these risks that we assess. So what we are doing essentially is just telling you the risk, and then on top of this we tell you that okay, you have a major or minor risk, but this is the work that you need to do to avoid these things to happening in the first place. And we also assess whether you have already developed or showing the symptoms of these disease existing through your lifestyle assessment and deep phenotyping that we do. So we have another question. Is there a system to match patients' medical profile with the published medical profiles of a study participants? Okay, so I can take this one, Elika. I mean, maybe you can add on to this. So yes, we can, we do take, when we say, so this is, we showed you the genetic part of our story, so to say. The other part, which is really important, is the deep phenotyping part, and which is actually true to wellOwise. No other company goes into that depth. So we do take a detailed case study or case history of each patient, which includes the lifestyle form, which is free and online available. Then there is almost like a case history interview with a one-to-one with the health consultant. On top of that, we ask for at least 2 to 3 years of their health records and a recent blood report. That would have a comprehensive list of all the traits that we are monitoring. And on top of that, we also ask for a 1 week diet diary, so to say, to assess what sort of dietary habits they have and things like that. So then all this is combined and put into a system, which we have not shown here today because of the time constraints. But yes, then we combine these polygenic risk scores on top of lay these deep phenotyping data, and then assess whether how a person will have or has the likelihood of developing particular disease or symptoms. And then based on the published literature, we go at the SNP level that, okay, maybe IL-10 is affected, and we can see that the immune related functions are affected in this person. So what can we do to boost his immunity now? What is known in the literature to boost his immunity? What supplements, what dietary interventions we can do at the moment? And then because we also do the clinical work in parallel to this, our consumer pipeline, we are trying to develop disease specific cohorts that will help us to further, validate our models in a disease-specific manner. Is that it, Elika?
Yeah, it's fine. Okay. There was another question.
Yeah. Okay, so have you created ethnicity Indian-specific SNP database yourself, or is it a SNP database available via NCBI? Elika, you can take this one. It's your hard
NCBI does not provide Indian-based ethnicity SNP database. But we have these ethnicity controls, right? And for each person in that control, we have their SNP profiles. So we can see, okay, out of these 100 or 500 or 1,000 controls, which SNPs which alleles are rarer the Indian ethnicity. So we have that from our controls, but there's also recently there was published Indian ethnicity based information for SNPs. Was it IGB? Saher, do you remember?
RGBI might have one as well, but there is one particular one, I'm forgetting it as well, Elika, at the moment.
Yeah,
but
we can look at, but you can look it up too. It was recently published. It was 2020, maybe summer or fall. Yeah.
Yeah, but the database that we are referring to here is basically we developed at our in-house database. That is basically a collection of 300 databases that were online, and it's a compilation of that. And then on top of it, we have put our own impact factors and functional and structural classifications on top of that
That's where
database.
ethnicity comes in. Yeah, but we have this base database pulled in from different other public databases like NCBI, DBSNP. And then we put in the ethnicity filters based on our controls.
Mm-hmm. Any other questions? I don't see. Is there any other questions? I think people can ask themselves. I have unmuted you all, so people can ask. I think that's about it. Okay. Thanks a lot, everyone, for your patience listening and all the great questions that came our way. And me and Elika and the rest of the team will be most happy to take any questions. Elika's email is shared on the webinar. It's elika@wellowise.com and mine is saher@wellowise.com. We will share it on the chat platform as well. Please feel free to contact us if you have any doubts or if you want to try our platform or need any help. Any clinicians listening to us are more than happy. We will be more than happy to share you our clinical pipeline as well. So today's talk was more focused on the consumer side of things. But yeah, our next sessions will focus on specific areas within clinical pipeline. For example, next one might be neurological diseases, cardiovascular diseases, and so on and so forth. Elika, do you want to say something? Sorry, I think there was somebody on the door.
Okay. That's wanted to let you guys know that you can find more information on wellowise.com. And yeah, I would encourage you to get the app and try it out, see what the free assessment looks like. And if you are interested, then we can add on the genomic analysis on top of it, which is a paid program. And it's pretty nice. You can track all of these things, your medications, allergies, symptoms, medical reports. So we'll have everything on file at one place for you.
Okay, thanks again, everyone, for attending today's session. Thank you all. Thank you, Elika, for such a good talk. Thank you.
Bye.
Bye.